Good morning all, I'm Conci, biomedical engineer from Politecnico di Milano, and today I'm going to talk about tissue regeneration imaged in vivo using a miniaturized window for intraviral nonlinear microscopy. Each time you develop a new drug or an implantable material, to reach the product launch you have to follow a well-known step of procedure that starts from the in vitro inspection and then reach the in vivo preclinical phase. In particular, that phase is based on tissue histopathological analysis made on experimental animals, as required by international authorities. This phase can last up to 4 years and is the most expensive and the most time-consuming into the validation process. That analysis required by standardized tests are ethically undesirable, expensive, and very subjective. As to reduce the number of employed animals in such kind of analysis, intraviral microscopy techniques have been introduced as an effective means to study the dynamic evolution of biological reaction in cell-living organisms, as to trace the same animal in different time points. To perform such kind of in vivo analysis, you need the imaging window, and recently scientists developed a new kind of miniaturized one that are based on scaffold that can be implanted in external window, for example the dorsal scaffold chamber, or subcut as a skin graft. These allow you to study the same animal for different time points, tracing the occurring of the reaction. Anyway, up to now, such kind of devices always require a direct percutaneous access and, in addition, allow you only to perform randomized repeated intraviral study, not responding to the requirement of the normative. Our goal is to develop a new kind of miniaturized imaging window, the so-called Microatias, that can be implanted subcut in a mini-invasive way. This is a multifunctional device that from one side allows you to guide the regeneration of the tissue, obtaining a microvascularization inside the device scaffold, and then allow you to repeat and localize your in vivo imaging, thus answering to the requirement of the normative. The microatlas is realized upon circular glass cover slip and is made by several microgrids. This can be disposed in a variable offensive and constituted by variable pore size, in our case of 50 micron. You can find some position landmarks will allow you to reposition the microscope field of view in the 3 dimension in different time point, allow you to trace the biological reaction occurring during the time in your animal. The scaffold height is in the range of 100 micron, and in addition, at the very edge of your device, you can find some spacer that allow you to couple your device with the target that you want to study in vivo. Now let's talk about the device microfabrication. Our device has been realized by means of the direct laser radium technique as the two-photon polymerization. This is a fast prototyping technique based on femtosecond laser source that allow you to reach a resolution beyond the diffraction limit. In our case, we exploit the biocompatible negative photoresist as the SZ2080 with a resolution of a quarter of micron. We realize such kind of lactase with the variable pore size. So, to validate our device, we decided to exploit this chicken embryo-living model with an ex vivo configuration. As to validate the feasibility of our imaging window, we decided to test it by performing the CAM assay. CAM assay is basically intended as a graft of our device upon the chordontoic membrane, the respiratory membrane of the embryo during its development. This membrane is highly vascularized and very reactive to the implantation of an external device, so we identified in relation to its growth a functional implantation and experiment window, thus implanting the device around the 8th day of incubation and reach until the 15 days of incubation as following. Then we demonstrate the cell repopulation inside our device by exploiting an ex vivo confocal fluorescence analysis. By exploiting confocal fluorescence microscopy, we demonstrated the feasibility of the Microatias in quantifying the cellular density inside our microscaffold with respect to a control tissue. Then, at the 3rd day of implantation, we perform a quantification of the cellular density, reaching a comparable value with respect to a control. Then, after 3 days, reached the 7th day of implantation, we reach a cellular density twofold greater with respect to a control that maintains a comparable value with respect to the previous time point. Then we demonstrate the cell infiltration in all the 8 of our microscaffolds. Then, as required by the normative in terms of quantification of the occurring reaction of an implant, we perform an in vivo label-free analysis of the blood vessel formed inside our device and the fibrosis generation in terms of collagen 1. Then a living chicken embryo was inspected by intraviral microscopy with a 2-fluorescence microscope. Then in autofluorescence, as you can see, we isolate neovascularization in control sample outside the microatlas and inside our scaffold. Then we proceed with the manual segmentation of our blood vessel and the quantification of the density of blood formed in the volume inspected. As you can see, what we obtain is a blood vessel density 3 times greater into the microatlas with respect to a control, obtaining a functional vessel network that can be quantified in different time points. To quantify the fibrosis rate, we perform label-free second harmonic generation imaging as to visualize the collagen type 1 inside the sample. As you can see on the right-hand side of the slide, we acquired control sample and microatlas 1 as to quantify the amount of directionated collagen type 1. What we obtain is a Gaussian distribution of the directionated collagen inside our samples, and in particular, we obtain a larger amount of highly directionated collagen type 1 inside our microatlas with respect to the control. In conclusion, what we obtain is a realization of a functional miniaturized imaging window for intraviral nonlinear microscopy, which can satisfy requests of normatives. For example, the ISO 10993 PAS-X in terms of biomaterial validation in cell-living animal. With our device, we can reduce the number of employed animal-do experimentation by observing and tracing the same animal sample during different time points, dramatically reducing the number of experimental animals inside the experimental group. What we obtain is a real-time quantification of the occurring reaction in terms of cell, neovascularization, and fibrosis. Then, for the future, we will be able to quantify the fatty infiltrate, improving our data by exploring the third harmonic generation, another label-free technique. In conclusion, I would like to thank all the team members, in particular Professor Manuela Raimondi from Politecnico di Milano, PRL project, and Professor Gilberto Chirico from Università di Milano Bicocca. And then, thank you all for your attention.